Hello friends, so we are back again with a new fungus. Well, this time it is in potatoes. So this is Clostridium fungus. So Clostridium is available, is found in the potatoes, is in the formation of pink patches or the pink molds in the potatoes. And these are actually this actually gets infected because of bad storage of potatoes. Nowadays these potatoes are stored in cold storages, and these cold storages are very bad. As I have said in earlier my videos, these cold storages are very bad. So this cold storages does not keep the potatoes well. So this affects this fungus, this Clostridium fungus. So this actually takes place and is found in the pink molds. And when you cut the potatoes open, you will see some pink molds. So later on in the videos, when we will be going to excavate these potatoes, you will see excavate this fungus, Clostridium fungus, from this potatoes. You will see how this Clostridium is found. It is formed in the potatoes inside. When you cut open the potatoes, you will see this Clostridium fungus. So this is very bad, and it causes a bad food poisoning inside our stomach. So it is regarded as a food poisoner by CDC lab Atlanta. So be aware of these potatoes. When you see cut open these potatoes and you see pink patches inside, immediately discard those potatoes. Immediately throw away those potatoes, because those potatoes are consisting of Clostridium fungus. This Clostridium fungus is very dangerous. It looks like a pink patches. So it is commonly known as the pink rashes in potatoes. The pink rashes. These pink rashes are due to this Clostridium fungus. So this we are going to excavate this fungus from the potatoes in the next part of the video, and be with us again. We will be seeing the mycotoxin and overall this mycotoxin is Clostrubin A. Maybe it is the only mycotoxin, or maybe some more mycotoxins are present in this Clostridium fungus. So later on we will be going in the fluorescent picture and the fluorescent imaging, and also the mycotoxin identification that we will be going on. So later on, let us begin the excavation of this fungus, this Clostridium fungus, and let us do the rest of part of the experiment, so that they can see the fungus and we will see how it looks like. So let us begin the excavation. So friends, we have started the excavation of the fungus. As you can see in the camera, and the below camera you can see it, and the other camera you can see these potatoes. So this is actually, you see here is the pink patches. So here you can see the camera is focusing, and as it is focused you can see these pink patches. So these are the Clostridium fungus. So this scalpel I am showing you. So these are the pink patches. So we are going to excavate these pink patches, and we are going to excavate the fungus. So we have 3 pieces of potatoes here, and all are full of Clostridium fungus. So one by one we are going to excavate, and let us see how much Clostridium we can take out. Just we are excavating the fungus from here with the help of potato host. Little bit host will also come, because otherwise if you do not take out the host you will damage the fungus. So we are beginning there. This potato has become very hard as it has been kept in the refrigerator. So it has become very hard. So anyhow we are excavating. We are just excavating the fungus. The potato has become very hard. So excavation is going on, and this part of the potato, it has come out. So it has come out, and we are just putting this in the host. So deep inside you can see the fungus has gone. So we are taking part by part and we are putting it inside. So this is over. So this part, so this is gone. So another potato we should take. As much as fungus we have to take, so that we'll able to see the growth of the fungus as what is necessary. So the potato has become very tough and it is very difficult to take out, because this happens when you put it in the cold storage. This actually happens to these potatoes. This actually happens. So these two are moderately, we have taken the fungus. Now this one is left. So this is the slice of the potato, and one thing happens with this Clostridium fungus. This does not allow the potato to get boiled even. You cannot boil the potato, and therefore you are deliberately you are going to get infected by this Clostridium fungus. So boiling, boiling actually does get rid of these funguses, but you are not allowed to boil, because this fungus will make the potato so hard. Actually, you see, the excavation is difficult. So you can immediately understand this so sharp knife. It is a very sharp scalpel. The scalpel is very sharp. If you touch your body, the scalpel will cut. So this is very sharp, and it is now making it difficult to excavate. It is very difficult to excavate this fungus. So why? So this is so hard. The potato has become so hard. So that is why it is due to the fungus. And this happens when you keep the potato inside the cold storage for a long time. So I have to cut open everything, and just cutting open the host, and cutting open the host, and we have to take the fungus. So this is another part of the fungus, and this part is left. So the host will cut off the host, and this is last part left, and this is the fungus. We have kept the fungus. Now we have taken the fungus. All the fungus has been taken out, and so the excavation is done. So here we will be going to add the Clostridium medium, the particular medium. Here we are using a very selective agar, selective culture media. As I have explained in my culture media videos, I am just breaking apart a selective culture media. Now this is Clostridium medium. Now this Clostridium medium will help to grow this Clostridium bacteria, Clostridium fungus, the Clostridium fungus far more easily. So therefore we will be going to take this Clostridium medium and take this Clostridium fungus to grow. So let us see. This is a green liquid, and lot of culture media is necessary because this is a good amount of fungus is there. So we are going to take, and this is enough. So the medium is taken, and the flask is ready for putting it into the incubator. So here we will keep this fungus for about 5 days period as usual. We have to keep this fungus for 5 days period, and we will see after 5 days. We will come back after 5 days, and we will come back after 5 days to see the fungal growth under TCM 400 microscope. So here we are ready to put this into the culture media. So this is our first part of this video. We are going to start with the microscopic examination after 5 days. So let us see what happens, how the fungus looks like, and what is the growth and all that after 5 days. So bye from now. Hello friends, we are back after 3 days with our Clostridium fungus. So actually this is a very fast growing fungus. As you found, it is already, you can see in the camera that the color has changed already. You can see in this camera the color has changed from green to light green or bluish green. So, and it has become very dense. So we are going to observe this fungus after 3 days. We have started this culture on 23/4/2022. Now today is 26th. So after 3 days we will be starting the culture. We will be observing the culture of this Clostridium fungus. So let us begin the observation. So for that, as the fungus has become very dense, we are going to take only 1 ml of this fungus. By the help of this pipette, we are going to take only 1 ml and 2 ml of the dextrosaline to dilute it, so that we can get a very clear view in the microscope, because this fungus is very dense and we need a clear view of this fungus. So it is very dense. As you can see in the petri dish, it is just sitting. So we need as you can see in the petri dish, it is sitting. So if you can see from here, so it is sitting. So that is the position. So you can see the position. So this is sitting. So we have to dilute this with the help of this. So around 1.8 ml of this Clostridium medium, Clostridium fungus. We are going to take physiological saline. This is our physiological saline. We are going to take 1.8 ml of this physiological saline and just dilute it, so that we can clearly see the things inside. So otherwise it will be a very thick mass, and as you can see, it is still a thick mass. So we are going to see the fungal matter. We are just diluting the fungus. So we are going to see the thick mass inside under this TCM 400 microscope. Now you will be able to see under TCM 400 microscope. So this we are going to see under TCM 400 microscope. So let us begin the observation under TCM 400 microscope. So this is the Clostridium Puniceum fungus under TCM 400 microscope. As you can see, the oocytes are there. These are the basic fungal matters. Now the oocytes are before your screen. So these are the oocytes, the microclostridiums actually. So these are the Puniceum oocytes. You can see many oocytes, collective oocytes are there, and after dilution by 1:2, it has become, or rather 2:1, it has become very clear. Now this the Clostridium fringes are there. So the development is very dilute now. So it is, though the Clostridium fringes are in front of you. These are the Clostridium fringes. These are the mature fringes. You can see stacks of fringes in front of you, and these are the running oocytes. You can see this is a oocyte, single oocyte, and these are the collective matured oocytes are in front of you. These are the collective matured oocytes. So the microscope needs to be focused, and you can see the oocytes inside. These are the matured oocytes. It's full of clustered matured oocytes in front of you. So this is the Clostridium Puniceum. and all is there. This is Clostridium Puniceum actually. This is Clostridium Puniceum. So these are the Puniceum mosaics. So after dilution, the collection has become less. So anyhow, to show you the clear picture, I have diluted these mosaics. So I have diluted this fungal mass. So this is the Clostridium mosaics, Clostridium Puniceum, and this is a very bad fungus, as you can see. And this consists, according to the database, you can see more mosaics are there. According to the database, if you see, we will see after the fluorescence study that it should consist of usiloxin, because it is consisting of viral DNA. So it should consist of the usiloxin. So that we will see after the fluorescence study. So this is all about the mosaics. So this is the Clostridium mosaics, Clostridium Puniceum mosaics, and that is the picture. This is a cluster, a very big cluster. You can see in the front, it is a very big cluster. You can see this is the microscope is being focused, and you can see it's a very big cluster. So these are the oocyte cells, actually the mature oocyte cells, and this consists of mycotoxins. So this we will see under the fluorescence culture after 2 or 3 days. So that we will see. And so this is all about the Puniceum, Clostridium Puniceum fungus. So this is all about. So this is the fluorescence image. As you can see, these are the Clostridium fungus under fluorescence microscopy, and you can see the micropellets and the presence of mycotoxin inside that, and it is Clostrubin A mycotoxin, it is present. So we will see in the UV spectrophotometry, but we can see the phases of the mycotoxin, the little bits, little dots, as you can see in the, these are the little dots. This is the Clostridium fungus. So these are the micro dots you can see. This is 400x magnification. This is 400x magnification. So these are the other parts of the Clostridium fungus. So you can see this microscope is focused. The slide has not yet fully dried, so therefore you will see some liquid part also. So this is the Clostridium fungus, Clostridium fungus. This is Clostridium fungus, Clostridium perennius. This is a Clostridium perennius, and this is the oocyte formation. The mycotoxin is in the outline border, also outer wall of this oocyte. This is the cellular wall of the oocyte is consisting of the mycotoxin. The magnification is very high, so you cannot see the full oocyte. So we are going to decrease the mycotoxin. Then this is the oocyte cell. This is the complete oocyte cell. You can see this is the border, the cell wall, oocyte wall of this oocyte. The cellular wall of this oocyte is consisting of the mycotoxin. Both has been impregnated with rhodamine or amine stain and IRCP stain. So this is the oocyte again. These are not the complete structure. The complete structure you will have to see under lower magnification. So the magnification is now getting decreased, and it is now 200x magnification. So you can see the total structure. So these are the oocyte formation. This is a very large colony coming in front of you. This is a very large colony. This is the very large colony, but inside there the mycotoxins are dead. The cells are dead. This is the dead mycotoxin, dead colony. This is a dead colony. The red patches only. These are the overstained patches. So these are not the mycotoxins. You can see. These are the formation of the mycotoxins. You can see it is in the rhodamine stain. It has caught the rhodamine stain. So these are the mycotoxins, the Clostridium fungus. You can see these are the other parts of the slide. You can see. So these are the red patches of the mycotoxin. The Clostridium mycotoxin has reacted, has got oxidized, and therefore the stain has become red. So this is a very beautiful prematured oocyte. This is a very beautiful prematured oocyte, as you can see in the front of the screen. So this will grow and become mature. So this is another oocyte. These are the other parts of the Clostridium fungus. This is a complete colony of a Clostridium fungus. This is the Clostridium fungus. So the magnification is 20x. So we are increasing the magnification again to 400x, and so you can see the colony better. We have to decrease the light. The light intensity is very much. The UV light intensity is very much. So we have to decrease the light intensity so that you can see the colony inside. This is a very small colony. It's a very small colony. So this is all about Clostridium fungus, the fluorescence image. So the Clostrubin mycotoxin has been revealed, and that is in the 664 nanometer. You can see in the right hand side, extreme right hand side of the screen. So that is the Clostrubin A mycotoxin. It's a very bad mycotoxin. So this is the condition of the pink slime of the potatoes. So thank you very much.